flavor of how drug development happens today. What are the kind of low-hanging fruit in which we are looking for tools? It's a very inefficient business, so any tools which will help us be more efficient, more productive, get more shots on goal, we're very open. So I'll talk a little bit about where AI is and can influence drug development. But I'll also end it by talking a little bit about my own expertise, which is in modeling and simulation of biological systems. So basically, simulation research for drug development. I'll talk a little bit about what I see are some of the limitations of AI and where we should tread a little lightly before sort of embracing all of the usefulness of AI. So that's kind of the talk today. So most of you probably have a rough sense of how drug development happens. I think the pandemic and everyone waiting for the vaccines to get developed was a big like PR for our industry. I think because everybody was following what was happening. When were the trials happening? When would the drug come out? So it was that's probably how a lot of you understand how drug development happens. So I'll walk through what the stages are. So, the first thing that we need to do is to understand what is the biological pathway we're trying to manipulate. Usually, there is a disease, there is some biology, some part of the network of the biology which is not working well. So, we want to put a chemical entity or a protein to manipulate that pathway. And when we do that, we hope that the disease symptoms. So, first, we need to get that idea. We need to identify what is called a target, and we need to identify what is a molecule which can engage that target and change. The next is we need to establish this is a real concept, and that often happens in animal models. So, what we call preclinical research. So, this can happen in mouse model, monkey, dog, in vitro, lots of interesting systems, in silico, which is some of what I do as well. And that is the proof of concept. And once we're convinced that this entity can be a safe and efficacious drug, then we launch clinical trials, often first in healthy people to make sure there's no big safety concerns. Then we look for efficacy. We keep on increasing the size of the trials till we feel like there is a statistical signal. And then it gets regulated, and then you're able to go buy the drug in the pharmacy. So this is kind of the way it is, idealized way in which it works. But as with most things, the real world is much messier. This process can be iterative. There can be a lot of stop and go. Something that we think works doesn't work, and we have to come back to the drawing board. Something that works very well for a long time suddenly has to stop because there are safety signals that we didn't foresee. All kinds of interesting things happen here. And all of this is really because, you know, biology is poorly understood. So, what is inside us, we don't understand as well as we could. So, there's a lot of open problems, but really, biology is very challenging. We don't understand all of it that happens in all layers. And more importantly, there's wide variability. A drug could work in one population and not in another population. So, all sorts of interesting things are happening. And because of which, as an industry, drug development is a risky business. It takes billions of dollars and, most importantly, decades sometimes to get an effective drug. And this is kind of a lost opportunity in terms of patients who are looking for these drugs, they're not getting it in time. So there's plenty of room to improve, plenty of more understanding to be gotten, plenty of innovation in drug development. And it's a very gratifying space to work because, it does at the end of the day improve. So I gave you a little bit of, like a hand wavy view and all of the, chaos and interesting things that can happen in drug development. Now I'd like to get a little more structured. These are kind of still flavors of the kinds of analyses that happen in the various stages that I was. So I spoke about a discovery early research stage, a preclinical, and translational is really anything that happens before human. How will it work in human? How does that translation happen? So there's an early research, translational, clinical, and the one important thing that distinguishes drug development from most other industries, it's highly regulated. At the end of the day, even if you have a miraculous product, the regulator of each geography, usually in the US it's FDA, but each geography has its own regulator. The regulator has to be convinced that the drug that you have is safe, efficacious, and a meaningful addition to the population. So all of that leads to regulatory approval, and that's kind of one half of a pharma company. All of our efforts are really focused towards how do we take it to the regulator, how do we get our drug approved. But that's definitely not the end of the story. On the other side is what we call post market evaluation, pharmacovigilance, real world evidence. Because even in the biggest clinical trials, there's about hundreds of people that take the drug, and we are convinced that the drug works well. But in a real life, when it goes out to thousands, hundreds of thousands, and millions of people, still need to keep an eye on what the drug does because the numbers are very big and we get signals. And these signals are sometimes positive. We find out that the drug is more effective than we thought in some populations. And sometimes the signals are negative. We see adverse events that we didn't see with hundreds of patients that we start seeing in millions of patients. So that means we have to go and evaluate. As drug developers, we also keep a close eye on what's already in the market because that provides our competitive landscape. If I'm coming up with a new drug for diabetes, I need to understand what else is there because however innovative or awesome my drug is or the mechanism is, if there is already very good medicines out there, it doesn't make sense to add it. So drug developers are constantly looking at the real world data to also identify what are underserved markets, what's a gap in. So, in all of these stages, these are kind of specific words and analyses that happen. And I think the flavor I want to give you is, at the end of the day, this is a highly creative and extremely interdisciplinary process. So, each of these analyses is done by different groups of experts, and all of it has to sort of come together over a decade to come up with something that has to happen. So, I'll just give you an example of one such analysis, right? So, what is called first in human dosing strategy. So, here, one important thing about a medicine is how much do you. I mean, there is an old, Greek saying by this Paracelsus, which basically says the medicine is in the dose. And I think every culture has its version of that. Something can be highly effective at a particular dose, but too less or too much of it is just like not the right thing to do. So, identifying what is the right dose to start a clinical trial is very important. But as you can imagine, if there's a completely new drug, you've only tested it in a petri dish, in some sort of in vitro setting, in a experiment like that, or you've tested it in a mouse or a monkey. So then, how do you sort of translate that? What sorts of analyses do you have to do? You do things called allometric scaling, where you go from what does it mean to dose a monkey 1 milligram? If that works, then what does it mean? How does the right dose have to depend on the efficacious threshold and the safety ceiling? So, safety and efficacy is always kind of the line that we walk in drug development. So, really doing that analysis is very important. Several scientists get together to do these analyses, and oftentimes it's different for different diseases. For instance, in a fatal disease like cancer, the safety tolerance is very flexible. People are okay to take a lot of safety risks because otherwise the patient's life. But for other conditions, the safety tolerance is much. So all sorts of interesting dynamics play, and that's kind of one sort of analysis that happens, sometime in between this 10-year process with, thousands of people contributing. So next, I'll talk about, given this diverse space, and I think this is a very wise audience. You're not like buying all the hype that AI is going to change everything overnight. But there are key aspects of this process where we're already starting to see contributions or meaningful changes in the way we do things because of AI. And today, specifically, I'll talk about three things which I find very interesting. The first thing, all the way at the left, is the design of the molecule. Like I said, what is the shape of the molecule which you're ready to put in human? Will it go and attack the biological pathway that we want it to? Will it change it appropriately? But more importantly, is it manufacturable? Will it stay the same at a certain temperature, at a certain pH? Is it transportable? Is it manufacturable at scale? So, all of these are kind of various aspects that you have to think about when you come up with the structure of a new medicine. And oftentimes, especially when you want to design a protein drug, that means searching a large solution. A protein is basically a set of amino acids, and when it all gets together, it can conform to a certain shape. And if you know what you want, what shape does that come to? So there's a large solution space that you can look at. And in the old days, we had to do it like laboriously, manually, with kind of very handwritten software. And the biggest change that AI has made is searching this protein space much, much. So you must all know about the AlphaFold Nobel. And today there are many, many drug companies that have products which have been generated by AI tools like this, which have gone pretty far. The Yeah, so this is a list of those things, and I'll come back to, we still have to take it with a bit of salt here, because a lot of these trials, the paper from 2 years ago, but a lot of these trials have stalled. Only some of these have gone ahead, which again speaks to, if you crack one problem, doesn't mean that you've solved drug development. There's just a lot of places where things can go wrong. The next thing, which I think somebody pointed out earlier, which is really exciting, is this is a complicated field. There are a lot of, hundreds of people working on it. So there is a lot of information that needs to be shared, information created by experts to be consumed by other experts. And the information comes from all kinds of diverse sources. Sometimes it's public data which is published in papers, which is freely available, often published by academics. Sometimes it's in-house data generated by the client in their compound. There you get a lot more detail. Sometimes it's competitor data, sometimes it's real world evidence. So, all kinds of interesting information has to come together to form a thesis which allows for the drug company to take the next meaningful decision. And all of this really kind of comes to a fever pitch when they go to the regulator, when they go to the FDA to ask for permission to do a clinical trial. So there are key meetings, and generating such documents is usually an intense process, taking, like any dozens of PhDs, months of their time to make sure we collect it all together, sit in meetings, convince each other, etc., etc. So as you can imagine, that's an excellent use case for GenAI and LLM tools, because generating these documents in a brisker manner, with understanding the riders that it needs a human in the loop to make sure we've gotten the details correctly and are able to convince each other, it can be like dramatically beneficial for this process. The last thing that I have there is in reverse translation. And here I mean a drug is already out there in the market, and we're starting to collect information from it. And this information too comes from very diverse sources. It can come from insurers' data, it can come from hospital chains' data, sometimes it comes from social media trends. All sorts of interesting things can contribute to that. And, again, collecting that data and making sense of what patterns that we see in sort of doing it at scale at a reasonable time, AI tools are starting to be pretty useful. I'll use this as a segue to kind of talk about the limitations of how far we can take this and where do we have to kind of make sure that we think about this carefully. So, this is an example. Recently, a big pharma client that we work with, they came and said, listen, we're seeing this interesting pattern in our real world tracking, in our pharmacovigilance data. We're seeing that diabetics are responding slightly different to this drug than the rest of the population. There's all sorts of interesting information about the gender, the diabetes status, the geography, but why are we seeing this pattern? So there's no mechanistic rationale. They were able to come from just that AI analysis of the data that we saw. They came to us, and we are a mechanistic modeling group, and they came and said, listen, like, we want to understand what's really happening in this data, so can you help us understand what's. I'll use this as a segue to kind of contrast what a white box world model type thinking can be for biomedical research, and then contrast and like make my case that we need both of these approaches to be really effective, drug development. So if you see here, this is, many people sadly are familiar with diabetes or have somebody with diabetes in their family. So this is a trend of plasma glucose in the blood that is measured over time when somebody is given a drink, let's say 300 grams of sugar in a Coca-Cola drink. So you can see the glucose, spikes and then comes back down. And if you're diabetic, the rate at which it comes down is different than if you're a healthy person. You kind of dispose of that glucose from the plasma much more effectively than a diabetic person. So when you do something like this, there is, several ways in which you can describe and understand this data. So the one thing is just what I said. Hey, the glucose is spiking and then it's going away. Another way to model this Another way to model this is purely empirically, where you can say, listen, there is an exponential decay. It starts off and there is a decay with a half-life of about 30 minutes, and that's how the glucose goes down. Another way to do this is to really understand what's happening. We know that glucose gets disposed away from the plasma because of insulin-dependent mechanisms. The better and more effective your insulin is, the easier you're able to shunt away the glucose. So there we go. So yeah, this is the sort of model that you can do, where you can say the rate of change of glucose that's happening depends on how much glucose you eat, how well your insulin clears it, how are the other mechanisms that. Once you have a framework like this to ask and interpret the data, then that allows you to kind of have knobs to understand what's happening. So suppose the insulin dependent clearance is poorer than I thought, what would that curve look like? And this allows us a point of view to understand the data. And to the point that somebody else said, it's not like this is going to be right and a black box approach is going to be wrong. Their thesis may still be right. The diabetics may still respond differently. But this allows us to have a hypothesis sort of approach. Of course, I'm not going into a lot of detail here. This sort of approach allows us to have an understanding of why that may be the case, and allows us to ask what if experiments suggest alternate hypotheses. So that's the kind of work that we do at Vantage. This is a little plug for the company I run. We're a small group of about 50 people working, most of us right here from Chennai. But because of kind of the effort and the expertise it takes to make models like this, we are able to work with kind of the best pharma groups in the world, because we're able to collect all this information with a multidisciplinary group of biologists and mathematicians who come up with these models and allow our collaborators to simulate trials, etc., before they, run them actually in the real world. So this comes back to kind of, I think, a point which was, emphasized by many of the speakers in the morning. And more than anything else in drug development, it's an intimate way in which you affect a human's life. And so the bar you want to cross to make sure that when you're, releasing a drug or making a recommendation that group 1 should get a different, dose than the group 2, then you better be clear that that is not motivated by bias, that's not motivated by a black box or understanding of what's happening, but rather you have a transparent way to understand why you're coming up with that recommendation, an explainable model where you say, listen, this is the reason why I think this is happening, and justify it to the best of your capability. And really, for a long time, there will need to be a human in the loop as we make, these AI-driven progress in drug development. But together, I think, as most drug developers I know are less threatened by all of these interesting things that may be happening and more excited, because like I said, even though it's a fascinating business, it can be a frustrating business. And being able to collect all this information, synthesize all this information, and transfer it in more effective ways is very, very welcome in our world. So with that, thanks again for having me. There are many, many papers on drug development and AI. This is from my corner of the world, which is quantitative medicine, model-informed drug development, and how the practitioners in our business see AI affecting our world. So thanks again for the opportunity. I really.
Thank you. That was a very interesting session. Actually, I was in pharma. So one quick question is with regards to the use of AI in drug development. Now you have a lot of novel therapies coming, be it CAR-T, xRNA, or even RLT. Right? I know for a fact, like you touched upon the personalized medicine, on how quickly you need to transport a medicine. For example, Pluvicto, which is a prostate cancer treatment, you need to take the radioisotope within 5 days to apply on the patient to treat. Same with CAR-T. So with regards to personalized medicine or new novel therapies like xRNA or RLT or even radioligand therapy, CAR-T or xRNA, I feel the relevance of using AI to identify the target molecules using AlphaFold and other things is much more applicable, because there you are talking about segmentation and it is not for wider population, it's more personalized. So where do you see the use of AI to look at the novel therapies where you don't need to really worry about pharmacovigilance, adverse event, real world evidence, but you look at the patient under treatment as the key
Yeah.
subject.
So excellent question, and I'll kind of flip it on its head and say the following, right? In principle, the scientific problems are very simple, very similar in kind of medicine at scale and personalized medicine, because you're eventually trying to do the same thing. But personalized medicine, pharma and biotech have been threatening to do that for a long time, but they'll never really do it because it's not a market at scale. So it requires a lot of work to actually do that. So anything which will lower the barrier to entry. So the reason big pharma and biotechs need so much funding is because they're trying to do medicine at scale. So anything that will lower the bar to make the analysis process and the scientific process easier is it can be very highly impactful. Scientifically, I feel they may be similar, but economically, the impact of AI on precision medicine may be higher for that.
Okay, Rukmini, thanks. Good presentation. My question is what is your thought on using AI or agentic AI or agents with pharmacovigilance testing? Today still a lot more are manually done. There could be manual errors coming. Have you seen that being proliferated across the industry? I mean, what's your thought? How much has it taken? 20%, 50%?
I don't know to give you a percentage because I don't understand that space well enough and it may still be pretty hard. But what I feel is, I think there is a lot more of that device revolution, right? Where people in faraway places which don't have access to doctors, both via telemedicine and via devices, are able to transmit their biomarkers, their readouts to large databases, which then become much more useful to be people put in a broader database in terms of AI. And I don't know enough to say that these are the niche markets of pharmacovigilance which can definitely be swooped up or made easier by AI, but I see those changes coming every like both devices, large databases, mining the internet in general for even things like social media boards, etc., where people talk about adverse events in a much freer manner than they would. So all of those become data sources to be absorbed by AI.
Thank you.
So where do you see the future for organ in a chip in terms of replacing the entire animal testing, and do you see a real future in that?
In some aspects, I think it can be very promising. In where I've seen it kind of be more useful is in liver toxicity. So liver toxicity is something which is relevant to every drug. So if you have a liver on a chip, then it becomes easy to test your drug on that, and it becomes a standard plate to test it for several. But overall, biology being as variable and complicated as it is, there are two layers, right? One, what happens in a tissue need not translate to what happens in an organ. It gives you the first signal, but what happens in an organ, you still, or what happens in an organism. What happens in an organ need not be what happens in an organism. So even if you get strong signals from a chip, you will still have to do some experimentation. So I feel like it will reduce the amount of animal experiments that happen, and it is limited by how complex biology is, but it will be it will kind of replace a lot of animals, as will approaches like in silico. So our claim as well is that, instead of doing multiple mouse experiments, if you even learn to optimize that in silico models, that will kind of make your life.
Drug development, implementation, testing would have been cut to a percentage of the time frame?
I don't know to answer that because I feel like, you may have thought it was slow, but in our business, that was the fastest a drug went from like concept to manufacturing and release. So I think in that particular case, maybe not, but in kind of those tables that I showed, I think several drugs have really accelerated that design phase pretty dramatically because of AI tools. The COVID vaccine in particular, I don't know to comment on that, but I don't think so.
So how about simulations that you were talking about? Now, how important is that in this entire cycle? If the cycle is 10 years, you're saying from discovery to whatever, to going out in the market, then does it, so where does the role of AI in simulation come?
So in simulations, which is not related to AI, the kind of stuff that we do.
Solving differential equations and so on, which you might want to do. Yeah.
Sorry, I can't.
Solving things like differential equations and so on, which I suppose are also models that you all use, right? Yeah.
And, I don't solve them, they solve them in the computer,
Yeah.
unlike the CMI folks potentially. But coming back to your question, right, I feel like a lot of technology that supports drug development is really about risk mitigation. Because if you have multiple options, running a simulation will tell you not to bother with option A and B and just go with option C and D. So risk mitigation is a big part of what a lot of what we do. I think we tried to come up with counterfactuals where, like, with simulation, the time spent and the money spent was much lesser than without simulation. But those analyses, in my opinion, are much harder to do. But day to day, in my experience, I feel like what we really do well is limit the solution space so that the company can focus their time and money on more likely.
Yeah, so you talking about ODs, I saw some professor was also asking about it. So when you're talking about ODs and linear models, wherein like say linear regression models and other things, wherein you have the coefficients, so there is no question of any being it a black box model or Okay, because so I saw a reference to the last slide, I think, wherein you were talking about transparency or those kind of things. So I was just interested to see if you've used any LLMs for pharma-related use cases. If so, any specific issue or any problem you face, because currently we're working with a pharma client.
I'm
and
happy
we're
to talk a little bit more. Basically, I was trying to draw the contrast between the ODE approach, which I think is open box
Yes,
and
yes.
hypothesis testing, and a AI black box model. LLMs, what we really use it for is, right now, is literature synthesis and documentation. So
Okay.
we use it like, almost without instruction or plan. I see several people in my company and others immediately going to Gemini and ChatGPT, because routinely our scientists will read like, dozens of papers a week, and this just makes that process much, much smarter.
Okay.
So that's what we use it with. And of course, we have problems. We have problems locating the right papers. There's a lot of hallucination there, but I'm happy to.
Okay. Thank you.
Thanks again.
Ma'am, one second, one second, one second. First of all, thank you so much for a very insightful session post lunch, and also to all the questioners. If I know still people have questions, she'll be here all available during the next IT session. You can please shoot your questions at that time. May we request Professor K.V. Subramanian sir to join us on stage? Okay, the one who had question as well. So he is here to felicitate with a token of gratitude. Thank you so much.